It's Mendel, so we're going to burn right through it real quick here. But Blackboard is just taking forever right now.
The wireless. The wireless.
Wireless in this building.
They're fixing it apparently, but yeah, apparently. It's almost dark for this. They have routers and everything.
They acknowledge that.
Okay, well let's just get started on some of the simple concepts. Is that on yet? or
Yeah,
not?
it's on.
Okay, it's on. So now we have the fun, exciting lesson of Mendelian genetics. We need to go over these basics just to make sure we're on the same page, and then we get into non-Mendelian genetics and things that violate Mendelian genetics and things that you're like, oh wow. Some of this stuff can be explained very logically. Some of this there's really not an easy answer to. And I'll just tell you, this is what logic would tell you, and this is what it should be. And logic is not followed, and reason is not followed, and we don't have explanations for it yet. Later in the course, we will get to answers for those things we can't yet get answers for. Right? That'll be section 3 of the course. So the basis, or the reason that Gregor Mendel was performing the work that he was doing is people would have like a prize ram. What's the opposite of a prize ram? You? Yeah. A prize ewe. They breed them together. Like this thing is, it's tall, has a lot of muscle on it, a lot of meat on it, so that when you're done harvesting the wool off of it, you get a lot of meat to feed your family. The wool, it just makes a bounty of wool. The wool is soft and really nice. And the same thing for this, it's just like a perfect match. You put these things together and you get a super lamb, right? But what they get is crappy lamb. That's this withered up, runty, really rough fur, patchy fur, doesn't produce much. I'm like, how can this possibly be? I took this incredible thing and this incredible thing, put it together, and got this garbage out. So they couldn't understand the patterns. And they'd say, okay, well, maybe if I take two crappy things and put them together, and every now and then, out comes this like super incredible creature. How can I take two awful things and put them together and get this amazing, wonderful thing? Okay? So these breeders, for hundreds of years, could not explain the patterns of how things are passed on. And that was the real basis of this. The economy was really agri-focused and livestock-focused back at the time. Your wealth was determined by how many goats or how many cows and such you had. And so there was a lot of interest in people trying to figure this stuff out. And, oops, that's that one. Mendel did something very unusual. He followed the scientific method. The scientific method. He said, I'm going to do this, I'm going to run this experiment, and I'm going to have it in a controlled manner. I'm only going to change one thing at a time. And when I get the data, if I don't like the data, if I say, I think this is the pattern, this is what I expect, if my data doesn't match that, I'm going to keep the data instead of throwing it away or squashing the ones that I didn't like and then not counting those. Like fundamental concepts. The data is what it is. And if my model doesn't match the data, I need to throw my model away and create a new one to match the data. And then test and see, does it work the next time, does it work the next time? So, he was really a scientist. And he did not start out as a scientist. So I'm going to have to redo this. I'm just doing this for the purpose of today. I'll get all this organized eventually. There. So, first thing is, humans have been engaging in genetic manipulation for thousands of years. Ooh, sorry. Oh, I shouldn't have done that. That's without these people there. But I want you to imagine, this is the starting material for a chihuahua. if you do selective breeding and you look for certain traits, after a few thousand years, you can actually wind up with Chihuahuas from the gray wolf. So, sorry about that for the people on the camera. All breeds of dog came out of the gray wolf, pardon me. All these guys. What's this? Chow, chow chow, greyhound, all these things. And basically, the evidence, it looks like wolves were beginning to be domesticated between 12 and 10,000 years ago. And what would happen is basically somebody got their hands on some wolf cubs, and they would raise the wolf cubs and try to maybe raise them for food or whatever. And the wolf cubs that would nip and bite and maybe scratch the person, those were not nice, and those were obviously the first ones to go into the stew pot. And the ones that were wouldn't bite so hard and weren't so mean and aggressive over the long term of their lives, those would make it to adulthood and be propagated. And so that eventually you get these things that become very wimpy, kind of wolf-like things that become even more and more wimpy, so you wind up with like a German Shepherd, right? It's pretty wolf-like in appearance. People have actually taken wild foxes and through very intensive breeding programs gotten very domesticated forms of foxes that you can now have as pets at home. And they have many of the behavioral and physical characteristics that we now find in dogs, and that is teeth that are not so razor sharp to cut you, very submissive in their approach to you, very human friendly. They're eager to approach and be around humans. And this has happened in a very short period of time, in many generations of breeding, maybe 50 to 100 years. Controlled breeding of grasses have created the cereal grains that we now use every day in our lives. Few people can go a day without relying on cereal grains. Let me tell you how mixed up people were back then. So originally, people would say, I have this incredible thing and this incredible thing. When I take the characteristics of this and the characteristics of this and I put them together, I blend them. And what I mean by blending is, if I have cream of mushroom soup and I have tomato soup, and I pour them together and mix that together, there's no way I can take this mix of stuff and then like pour it here and all my tomato soup goes there, pour it here and all my mushroom soup goes there, right? They're forever mixed together. So this idea of the law of blending is thought to be true. And the other one was the law of unequal contribution. That is, the females are the receptives, and the male deposits what the next generation is going to be, and the female simply nourishes that and gives birth to that. Such that we would expect the progeny to basically carry most of the characteristics of the male, and they might be tempered a little bit by some of the characteristics of the female. And over time, some of those characteristics might get imparted in the line, if that line continued for long enough. And this idea of unequal contribution, that the males contributed most of the characteristics of the next generation, was so common sense that when microscopes were first being invented, people would look at microscopes. And here, don't ask me how this guy got his hands on some human sperm, but this guy got his hands on human sperm, looked at him under a microscope, and sketched out what he saw, and he very clearly saw the preformed baby in the sperm. It's called a homunculus. And this person even predicted, at some point we will develop very, very high-powered microscopes that will allow us to focus into the reproductive area of this child, and if this child is a male, we will actually see a homunculus in this homunculus, such that we could keep going down and down and down, and we could keep looking at the smaller, we could see all of the generations of this line are contained in this, and they just get smaller and smaller and smaller homunculus to the homunculus, right? Does that make sense? And of course now we have EMs, that's what we found, right? Electron microscope. Right? So that's some of the misconceptions or the common sense thinking that's the environment that Mendel was in when he was operating. This is actually a photograph of Mendel. There are a couple images of Mendel. Photography was coming out right there at the end of his life. I'll bet he did his main work in the 1860s, I'm going to say. I don't remember how late he lived, maybe the 1880s, right when photography was first coming out. He made the most important fundamental, laid the foundation for genetics. He published this in a journal. And there were probably, I'm making a number up here, there were probably 50 copies of the journal that were published. And most of those just kind of went filed in libraries, and then eventually the library said, oh, this is not an important journal, nothing important is published here, so they were gone. And so this finding never made it out into the scientific world. People said, oh, he's studying peas. Great. And the farmers would be happy. They didn't understand what he found was a fundamental law that goes across all life on Earth. It was in the 1900s that people running experiments figured out what he had figured out 40 years before, and they were like, awesome, I'm going to be incredibly famous because I did all this great work. I just need to go back and do a literature search and show how I came to put together my thinking. I need to explain the history of why we've now come to these incredible findings here in the early 1900s. They went back and they were like, what the hell, this guy found this 40 years ago. And I can't just claim it as mine because I have to document the history of what happened. And so then we discovered how important Mendel was, and he was dead by then. He worked in his father's orchard, and he worked as a substitute teacher, and he basically took the teacher certification test so he could be a full-time teacher, and he failed. And he failed on the nature and history portion of the exam. So he said, well, apparently I'm weak in that area, so he went to college, which just didn't happen back then, to learn about nature and history, to shore up his weakness, and it actually became his strength. While he was at college, he learned to look at the world and try to organize it and find patterns and then model those patterns. So he learned to start thinking like an engineer. Does that make you feel good? He published, it was 1866, he published his finding, Experiments on Plant Hybrids. That was the name of it, and it was published in the Proceedings of the Brune Society of Natural History. How many of you have read that one? So I can remember when I was in college, a friend of mine was getting his PhD in biology, and he published one of his papers in like the Minnesota Prairie Grass Journal. How widely read do you think that is? Right. Not widely read even in Minnesota. That was the right fit for his paper. It's the right spot. The people who were interested in his work were reading that journal. The 20 people on Earth interested in that, right? He couldn't get his work published elsewhere because people didn't understand the significance. They go, you're breeding peas. You learned about peas, great. That needs to go in this obscure little journal over here. He sent a copy. You ever heard this guy, Charles Darwin? So he was continually plagued about, he explained this idea of natural selection. And people would come back to him and say, great, okay, so there's genetic diversity and then things with the right type of genetic pattern fit into different niches and advance, and that's how you get new species. Where does that genetic diversity come from? How does that stuff get mixed around? And Darwin couldn't explain it. If Darwin had read this paper and comprehended it, he could have satisfied that and he would have been much more famous than he is today. Because this is a continual weakness in what he couldn't defend that position. Mendel mailed a copy of his paper to Darwin, and when they eventually went through and cataloged Darwin's papers in his office, this is only about 50 years ago, they found the letter from Mendel in an envelope that had never been opened. If you'd opened it and read it, you would have had it. Anyway. So don't throw away that credit card offer. You never know what's in there. Just kidding. Why did Mendel succeed? He used a model organism. And if you don't know what a model organism is, you need to learn this. A model organism is some creature that is representative of a whole other group of organisms. So we use mice. We run experiments on mice. And the way mice respond to feeding, if we feed a mouse a whole bunch of cancer compounds, and a mouse gets cancer, we say, okay, we're going to say, probably if we feed those same compounds to a human, the human's going to get cancer. So we try to extrapolate and say, okay, what happens there applies in other circumstances. So he used a model organism, and he used peas because that's what he was familiar with as plants. But what he was modeling was not pea plants, and what he was modeling was not plants. What he was modeling is things that sexually reproduce. Plants, animals, fungi, most of the things we think of as life. If he had been able to make that distinction clear that this is a model that represents all important life that we think of, all these livestock and all of our crops, he would have been able to publish somewhere else, but he apparently couldn't make that argument properly. A model, why do we study model organisms rather than humans? Well, I'm not interested in my kid being rounded up to be in the subject test to see if this compound is cancerous. How many of you would like to go out and have a couple kids so that we can put those into studies, feed them a bunch of stuff, and, hey, is this red dye that's in our food, is that a cancer-causing compound? Let's have your kid be the test and see if it is. Sound good? Who's up for that? We'll give you $100 a month. So we choose a model organism that doesn't complain, right? It's okay if a mouse gets cancer, maybe, unless you're like, what is it, PETA? People for Ethical Treatment of Animals, or some people would argue against it, but I'd rather we learn in mice and then say, oh, we prevent human disease as well. Model organisms are important. You need to be able to control the variables. You need to control the crosses. I have this known and I have this known. I don't want random mating to occur. I want to control, I want to breed this with this and not with this. So the mating has to be done precisely. Mendel was able to do that. So if you remember your whorls of the flower from Bio 2, you got your petals out like this. You got the carpel that goes up like this, and that's basically the female portion of the plant. And you've got your stamens out here where the pollen is made. So what you can do actually with peas is when the flower starts opening, the pollen isn't quite ready yet. Or you could actually open the flower a little bit early, and you can take scissors and you can cut all these off so that this thing can't self-pollinate. Once you cut all of the pollen-producing structures off, you actually took little bags, little cloth bags, and you would tie it over and seal it up tight. So that pollen from some other plant wouldn't be able to come and fertilize this. And then when he was ready, he would say, I want to mate it with number 17, plant number 17 over here. He would take the cloth off of flower number 17, and maybe this one he left the stamen on, and he would take the pollen off of here, and he would walk over and he would physically put them on here. And he controlled the mating precisely. Then he put the bag on and sealed it back up. Now, when you progeny off this, he knows this is the mother, and that was the father. Does that make sense? And the other thing is you can work with large numbers of progeny. So, for example, elephants have a progeny once every 2 years at the best. So if you want to do a study in elephants, to get yourself a large population of elephants is going to take a while, and then they need to grow like 20 years before they're reproductively viable. It's going to take you a long time to do your study in elephants. But mice, every 3 months you get a new generation of mice. So you could rapidly turn them over and you can test the effects over multiple generations. With peas, he was actually able to get 2 crops each year. You could plant seeds, grow them, and get the harvest in the spring. And then in the summer, you could plant those seeds and get another harvest in fall just before winter. So you get 2 generations in each year. Yeah.
How long did he do this study?
He spent a couple of years just doing some breeding experiment, or just showing, controlling the process to make sure he had pure strains. Probably did that for 2 or 3 years. And then he probably spent maybe 6 years doing his actual experiments. That's it. That's a PhD. So a long time. 10 years probably. $10 years is not a long time. 11 years I'm retiring. So 10 years is not long. Please tell me that. I get to teach general biology 2. I've taught it 16 times. I can almost blow my head out. I've taught it so many times. I get to teach it 22 more times. Awesome. I'll get even better at it, do you think? All right, let's move on here.
Maybe something will change. Something fundamental in our ideas of biology will change. And then you'll have to.
I'll get to what?
You'll get to
Change the course?
change your.
Let me tell you what's going to change. This camera being on up here, be very careful. The course is going to change into a studio format, and I will have the amazing opportunity to change my teaching style.
Oh, amazing.
Okay, Mendel studied discrete traits. What do I mean by discrete trait? Peas, the flowers are either purple or white. There's no light purple, there's no like slightly tinged white. It's either white or it's purple. One or the other, it's categorical. Human height is not a discrete trait. We took all the students in minds and lined them up by height. You wouldn't say, we have the small group and we have the tall group. You have a gradient in between. You get every possible, essentially any possible height in between, right? So he studied things that were easy to classify. And some people would say, oh, well, the fur here is soft, and the fur here is soft, and the fur here is not as soft, so we're going to call it not soft. And you're like, well, can you quantify that? Yes, it's not as soft, right? So it's this kind of squishy definition. So he was working with things that were very clearly defined, and he did seven different traits. He actually did more than that, and only 7 made sense, and he threw away data. We'll talk about that later. Maybe we'll talk about that later. Some traits didn't follow this, and so he said, I'm only going to talk about the traits that worked, and I'll figure out the details on the others later. So these are the traits that he studied. Did plants grow tall or short? And there was no in between. Flower color, position of the flower. Is it axial? Is it grow from an axillary bud, or is it on the terminal bud? Is it an apical meristem that turns into of the flower? The color of the seed, the shape of the seed. Is it round, or is it all shriveled up and wrinkled like it's been in the vat for too long? Is the pod green or yellow color? And is the pod kind of full and puffy, or does the pod kind of squish itself around so that you can actually see the outline of the seeds? So those are the traits he studied. You don't need to know the traits. You need to understand the patterns. Something else he did is he used pure breeding lines. People go, oh, I got this prize ram from Joe over there, and the prize ewe from Sally over there, and bred them together. No idea what the breeding was ahead of here, and what the breeding was ahead of here. There's no idea. This may have been a really crappy breeding line, and somehow this awesome thing got spit out. So what Mendel did is he spent several years saying, I want to study things that have, I want to study plants that grow tall, and I'm going to inbreed those guys. And if I ever have a plant that spits out a short one, that thing's not pure breeding, and I throw that away. And eventually I'll get down to organisms that only produce tall plants. Now I know this thing is a pure, through and through, it's a purebred tall plant. I think he did like 7 generations to assure he had pure breeding lines. So you see up here? He controlled every variable he could, anything he could think of. So, when we're talking about plant size, if I grow a plant in shade, is it going to grow as tall as a plant that I grow in sun? No. So he made sure If I plant the short ones here, I'm going to measure the amount of sunlight they get. And the tall ones will be planted in a spot that gets the exact same amount of sunlight, the same amount of water, roughly. And so he was, any variable he could try to control, he did what he could to control it. I mean, this is common sense science stuff. But back then, people didn't do this. He generated a large amount of data, and he used all the data. Okay, I did this cross and I got all these things, and I don't like that I've got 5 of these things that don't make sense to me. I'm not going to throw those away. I'm going to use all that data, and off I go. It's funny. I used to teach in this genetics course. We used to do Drosophila crossing, and I can remember one time students had set up this cross and they got those results, and I knew they're supposed to get like 3 to 5 weird-looking Drosophila, which are screwy. And when they filed the report, they didn't have them. They didn't have those organisms. I was like, well, what happened? Why didn't you get this? You know you were supposed to get some of those. And eventually, after cajoling and kind of working on the students, eventually one said, oh, well, you know, they're on the paper and they didn't look right, so we're just going to crush them. We figured that it was an anomaly and an aberration, and we didn't know what to do with it, so we squished them on the paper and then threw the paper away. And then we had this nice clean data, and we drew our conclusions, which were wrong. Their paper was wrong. Is that funny? I know you guys would do something like that. That was ancient history, you know, like 4 years ago. The other thing is he ran reciprocal crosses. What do I mean by reciprocal cross? If the dad has purple flowers, and is pure breeding, and the mom has white flowers, he would do that cross, see what happens. And then in a separate part, in a controlled experiment, he'd have the dad with white flowers and the mother with purple flowers, and you do that cross to see the results. And because he did reciprocal crosses, what he was doing, he was saying, who contributes most of the characteristics in the next generation? Is it the dad, like everybody says? If so, in this case, we'd expect purple-flowered organisms, and here we'd expect white-flowered organisms. So he was actually testing the law of unequal contribution. And what he found, first generation every time, is it didn't matter which parent was which. There was a dominant trait. This came out purple. That was kind of a bad stink. He was probably going against the church at the time, too, and saying that it's not the males. The females and the males contribute the same. Careful about that stuff back then. That'll get you burned, right? So he did self-crosses to get true breeding strains the first couple of years, and then the next, for each of those 7 traits, he examined inheritance of just that one trait. He did that for, I don't know, 3 or 4 years. So what he would do is he would cross tall with dwarf, and so he's looking at only one trait, so that's a mono-hybrid cross, right? One trait. So this is what it looked like. Yellow peas and green peas, and crusted together, and they were all yellow peas. Green trait was lost. So when he blended them together, apparently yellow dominates over green. And according to the law of blending, green is forever missing out of this population, right? But when he had these guys inbreed with each other, in the next generation, he got some green ones back out. So right here, he gets back out a discrete trait. If blending occurred, these would be a little bit darker yellow or lighter green, and you would never get a distinct, sharp green back out. And most people in the old days, they would do this cross and they'd say, oh, my experiment didn't work out right, this cross didn't work out right. They wouldn't look for another generation down to see what happened. So he actually took these out multiple generations. He went beyond the parental, beyond the first, what they call filial, beyond the second filial, he went down to the third filial, and he did additional generations after that. And these are the actual data from his notebooks. He had 6,022 yellow seeds, 2,001 green seeds, a 3-to-1 ratio. That's actual data from Mendel. So, somehow, green must exist in here, but it's hidden. The green character is still in that population somehow. So, first thing is blending did not occur. There must be 2 types of yellow seeds. There must be yellow seeds here that are yellow, and that's it. But there must be yellow seeds in this population that can somehow produce green. So, based on this, each seed must have a dominant form of the inheritance and a hidden form of the inheritance, which we now call recessive. So there's 2 units of inheritance, not just one. And we know this, we know this now as being, we're 2N, right? We have 2 sets of chromosomes. Also, in this simple experiment, because the reciprocal process followed the same pattern, he could throw away that unequal contribution. So this is how we look at it now. For those who don't know, you're dominant. When you write the dominant trait, yellow is dominant, you write that in the capital. The recessive trait, so it's yellow is dominant, so you use Y for yellow. So the recessive is still this same letter, but it's small. So this first cross he did, a parent would contribute, this parent would contribute a dominant and a recessive, and the dominant wins out, so these things would look yellow. But when we self-cross this, we're basically doing this. We can get this guy can match up with this guy. This guy can match up with this guy. This guy can match up with that guy. Or this guy can match up with that guy. There's 4 possible outcomes. This one would be green. This one's yellow, and this one's yellow. 3-to-1 ratio. I hope you've gone through this and you're just bored out of your skull right now. I'm going to go through this very fast because you should have done all this before. This is the law of segregation. 2 copies of a gene are inherited at fertilization. When gametes are formed, when pollen and egg are formed, those copies separate, and how they separate is random, or they separate into their separate gametes, and the gametes that come together, which gamete chooses which to match up is random. And that's the story we're going to stick with for right now. So, to test, we can test the law of blending. We can test the law of equal contribution or unequal contribution. And we can test the law of segregation. I probably spelled segregation wrong there because I don't have a computer screen on here. We can test all of these just by following one trait in monohybrid cross. He actually did run an F3. If we take this population, which I've got here, and I self-cross all of those guys together, these guys are only going to produce yellows. These guys will actually produce in a 3-to-1 ratio. This is basically the same. These guys will produce in a 3-to-1 ratio, and these guys will all be pure breeding green. So if we take this population, I would expect 1/4 of these guys, 1/4 of this population would be all pure breeding yellow, 1/4 would be all pure breeding green, and half the population would give me this 3-to-1 ratio again, right? So he ran that out again and it worked. And he repeated that for another one, for an F4, and then he went maybe to F5, F6. He bred those all out for one time. Pretty cool, huh? 1906 is when people were all starting to discover for the first time, but it was actually redoing the work he'd done. They weren't aware they were redoing his work. And they actually, it was a researcher named Punnett, and there was another one I can't remember, but they would start to illustrate this on the Punnett square. If you're in this class, you've done a Punnett square before. If you haven't done one, stop by, I'll show you how, but I'm not going to teach you how to do a Punnett square. You need to know how to do that by now. Okay? So, just want to make sure we're all on the same page about these terms. Genotype means the actual sequence of the DNA, the genes that you possess, the versions of the genes you have. So, phenotype is what an organism looks like. So, this organism here, its phenotype equals yellow. Even though it has this, it looks yellow. The appearance to the eye is yellow, phenotype. The genotype on this one is actually, it's a heterozygote. It has one copy of each. This guy is little y, it's homozygous. It's a homozygote. Only one kind, and it's green. This is homozygous. It's only one kind, and it's yellow. Okay. You should know those terms by now. What's the genotype ratio here? 1 to 2 to 1. So we have one of these guys, two of these guys, one of these guys. This basically is that, right? So the ratio of genotypes here is 1 to 2 to 1. What's the ratio of phenotypes? 3 to 1. There's 3 yellow and 1 green. Okay. Then Mendel did what we call a dihybrid cross. I don't remember the example I have here for this. But he was testing the law of independent assortment. And I'm going to tell you, he got really, really lucky. He looked at a bunch of different traits, and basically what he was saying is, if I have something that is yellow, yellow, and I'm going to do tall, tall, so this guy is green and dwarf, am I going to get everywhere the Y and the T go, the capital Y and capital T, is it always going to sort out that way? Are these guys linked together? Right? So that's what he was testing. Or could I get an individual that's like this? Does that make sense? Do the genetic code for seed color segregate independent of the genetic code for plant height? Or are they stuck and linked together? So he tested that. To do that, you need to do what's called a dihybrid cross. He tested the law of independent assortment. Who has never done a dihybrid cross before? Okay, great, hold on. I'm not going to run through the Punnett square on this. I'm going to tell you my little trick. What kind of cross do you need to run to test the law of independent assortment? This is my little trick from college. Remember this? I'll give you guys. I know what type of cross to run to test the law of independent assortment because di is 2, and this is 2 words. Yes? A monohybrid cross is 1, and that tests the law of segregation, plus these other things. There are 2 reciprocal crosses, right? That's how I was able to keep it straight. And then I said, well, what's the purpose of a dihybrid cross? Okay. Shows that you got these 2 traits. Does that help? It's my little trick. Is that a mnemonic? Is that what that is? So here's just the basics. We've got round seeds. Since round is dominant, wrinkled is a little r. You run the cross, and we've got all these different possible combinations for the gametes. You draw that in this big, huge dihybrid cross here. Does anyone know the phenotypic ratio of a dihybrid cross? 9 to 3 to 1, yes. So, in this particular cross, we've got round and yellow. And we have wrinkled, green. Oops. So the possible combinations are round and yellow, round and green, wrinkled and yellow, and wrinkled and green. That's all the possible mixing combinations you could get of these guys. So this is one of the originals, and this is one of the originals. So that these are called recombinants. The traits have mixed up. If you lay out your dihybrid cross, you have 9 of these, you have 3 of these, 3 of these, and only 1 of this original up here.
Wouldn't that be if you had a cross between both heterozygotes? Because in this case, how would you get to a sensitive if
Sorry.
one is completely dominant?
This is your parental. When you cross this, you're going to wind up with this. Right? And then when you self-cross these guys, that's your first filial. We call that the F1. Now when you self-cross these guys, this is your F2. Sorry, I skipped over that. It's your, so these are the grandparents. These are the parents. And these are the grandchildren. Yes. So I was looking at 2 generations down. You get a 9:3 to 1 ratio. That's the phenotype. What's the genotype? I don't know. It's messy. Well, there's 1. Well, it's going to be something 1. And this is going to be 1. That's this guy. That guy and that guy are the same. There's a 2, which means that guy's an oddball. I don't know. I don't want to work through it. See how I'm doing that, though? I'm an exam. Now that I'm putting it up here, I'm probably going to ask you an exam. Right? I'll give you guys the incredible opportunity of an exam. Does anyone have other questions about dihybrid crosses? This very simple diagram basically shows this. Oh, here we go. I was just 9 to 3 to 3 to 1. Okay, we don't have time to do this, but I'm going to show you how you can run a octo-hybrid cross. How you calculate the ratios of the octo-hybrid cross when we come back next time. Okay? Thank you very much. Sorry it was late. I'll get the stuff online.